Why are we still here while Neanderthals and Denisovans vanished from the Earth? It's one of the great mysteries of our origins, how Homo sapiens managed to survive when our closest relatives didn't. Recent discoveries suggest a surprising clue, a tiny change in our DNA in an enzyme called ADSL may have played a crucial role in shaping our behavior and our brains, a microscopic difference that could have influenced how we sought resources like water and ultimately how we endured. If you find this fascinating, don't forget to subscribe for more insights. Around 600,000 years ago, the ancestors of modern humans branched off from the lineage that would later give rise to Neanderthals and Denisovans. For thousands of years, these groups shared a common evolutionary path until subtle genetic changes began to set Homo sapiens apart. One of the most intriguing of these changes was found in the enzyme known as ADSL. Within its chain of 484 amino acids, a single swap occurred. At position 429, alanine was replaced with valine. On the surface, this appears to be a trivial mutation, but this microscopic shift created a version of ADSL that is unique to Homo sapiens and may have reshaped our evolutionary destiny. ADSL, short for adenylosuccinate lyase, plays a vital role in the production of purine, one of the essential building blocks of DNA and RNA. Without purine, cells cannot replicate or repair properly, and our nervous system would not function as it does today. What makes ADSL especially fascinating is how even a small alteration can ripple through biology and affect behavior. In modern humans, when ADSL is completely absent or severely impaired, it leads to striking consequences. Behavioral abnormalities, such as hyperactivity, impulsiveness, and aggression. This highlights just how central the enzyme is to normal brain chemistry. When scientists compared the Homo sapiens version of ADSL to the one carried by Neanderthals and Denisovans, they found a key difference. The extinct relatives had a more stable, more efficient form of the enzyme. Paradoxically, our less efficient variant is the one that survived. Instead of being a weakness, it may have subtly changed how our brains processed signals and responded to environmental challenges, providing the groundwork for behaviors that would later give us an edge in survival. To investigate how this genetic difference might influence behavior, scientists turned to mice. They created two groups, one genetically modified to carry the human version of ADSL, and another control group with the natural mouse version. The goal was to see whether this subtle enzymatic change would produce measurable differences in behavior when resources were scarce. The test was straightforward but revealing. Over the course of 12 days, researchers gradually restricted the animals' access to water. After this period of deprivation, water was made available again, but only if the mice responded to a combination of light and sound cues that signaled the location of the dispenser. This setup mimicked a scenario where survival depends on detecting and responding to environmental signals in order to secure a vital resource. The results were striking. Female mice carrying the human variant of ADSL consistently visited the water station more often when thirsty compared to their unmodified counterparts. This was not just random movement. The modified mice seemed to be more attuned to the signals and quicker to act on them. Such a small molecular change had created a measurable difference in how the animals sought out water, hinting at a deep evolutionary link. What could this mean for our own evolutionary story? The researchers suggest that the less efficient ADSL variant may have given Homo sapiens a surprising advantage. In environments where water was scarce, individuals with this mutation may have been more alert to signals of its presence and more persistent in seeking it out. Over time, even small behavioral differences like these could translate into better survival and reproduction rates. While Neanderthals and Denisovans carried the older, more efficient enzyme, it might not have provided the same behavioral flexibility. For Homo sapiens, the mutation may have fine-tuned motivation and responsiveness, traits that could have been crucial in unstable environments where every drop of water mattered. Digging deeper into the modern human genome, scientists found that the story doesn't stop with a single amino acid swap. In fact, at least 97% of living humans carry a cluster of genetic variants in the ADSL gene. These variants make the enzyme even less efficient at expressing RNA, potentially amplifying the behavioral effects first triggered by the alanine to valine substitution. This pattern strongly suggests positive selection. In other words, these weaker versions of ADSL were not just random quirks that spread by chance. They were favored and passed down because they offered some kind of survival benefit. The fact that this gene sits in a region of the genome known to be evolutionarily favored in Homo sapiens further strengthens the case. It may seem counterintuitive that inefficiency could be adaptive, but evolution is not about perfection. It is about advantage in a particular context. For our ancestors, subtle behavioral shifts linked to this gene may have been exactly the edge needed to outlast their rivals. The discovery of the first full Neanderthal genome in 2010, followed by the Denisovan genome 2 years later, allowed scientists to make direct comparisons between our DNA and that of our closest extinct relatives. The results reveal a complex picture. Many of the changes that distinguish modern humans are tied to how our brains develop and function. But ADSL alone cannot explain why Homo sapiens endured. Our survival was likely shaped by a convergence of factors: adaptability to shifting climates, social cooperation, resistance to diseases, and innovations in tools and culture. Neanderthals and Denisovans, despite their strengths, lacked the same combination of traits. Chance also played a role. Evolution is not a predetermined path, but a tangle of opportunities and accidents. Still, the persistence of our unique ADSL variant suggests it contributed to the resilience that ultimately left Homo sapiens as the only surviving hominin species. As compelling as these findings are, scientists caution against drawing direct lines between a single mutation and human destiny. The experiments were conducted on mice, not people, and while they offer valuable clues, they cannot capture the full complexity of human behavior. Intriguingly, the changes in water-seeking activity appeared only in female mice, raising further questions about sex-specific effects that remain unexplained. Moreover, more than 80 other amino acid differences separate Homo sapiens from Neanderthals. A DSL is just one piece of a much larger puzzle where genes, environment, culture, and chance all combine to shape who we became. A single amino acid change in the enzyme ADSL may seem insignificant, yet it could have nudged Homo sapiens toward survival while other human species disappeared. This tiny genetic twist highlights how evolution works through subtle shifts with profound consequences. Many questions remain, but each discovery brings us closer to understanding why we, and not our cousins, endured. Stay tuned. Subscribe for more explorations into our human story.